Good afternoon, everyone, and welcome back to yet another Ifakara Talk. Happy New Year, and this is our first stop today, which will be a two-part series that seeks to explore the current state of tuberculosis and neglected tropical diseases. This conversation today will be in partnership with our collaborators from the Swiss Tropical and Public Health Institute. Part one, where we'll be discussing about neglected tropical diseases, specifically the impact of schistosomiasis and soil transmitted Helminthiasis on sustainable development goals. Particularly for this talk will be Professor Jürg Utzinger, who is a distinguished figure in the field of epidemiology and currently serves as the director of the Swiss Tropical and Public Health Institute, and also a professor of epidemiology with the University of Basel. He has been actively involved in transnational global health research consortia that have fostered international collaborations, leading more effective and sustainable health interventions in countries such as China, Cote d'Ivoire, South Africa, and Tanzania. His work has significantly contributed to raising awareness and advocating and advancing the control and elimination of neglected tropical diseases. Professor Utzinger has been a part of various global health policies and efforts, including the London Declaration of Neglected Tropical Diseases, which aims to better address these diseases and impact on vulnerable populations. Professor Utzinger, that's a wholesome biography to yourself. What inspired you into neglected tropical diseases?
First and foremost, thank you very much indeed for having us here. Our distinct pleasure to be with you now for this afternoon session. I'm also here together with colleagues. And the neglected tropical diseases are diseases that attracted my attention because it's really focusing on neglected communities, neglected issues. And that's where I feel with concerted efforts, we can make a real difference.
Absolutely. So please tell us more. What is the current state of research within this space, and what is expected of us if we ever aim to achieve the sustainable development goals?
If you don't mind, I would like to set the stage with a couple of slides to just frame the context, and then, of course, more than happy to answer on your very specific questions.
Karibu sana.
Asante sana. So what I would like to present in the next 10 or 15 minutes is a bit of the work that is done at the Swiss Tropical and Public Health Institute to better understand the epidemiology, the control of neglected tropical diseases. But to do that first, I would also like to give you a very brief overview of where we are and also the long-term partnership we have with the Ifakara Health Institute. So basically, the Swiss Tropical and Public Health Institute is based in Basel, Allschwil, and Basel in Switzerland. We are in a brand new headquarter, and we have cutting-edge laboratory facilities, an open space, offices, but most importantly, then also a network of partnerships in basically in disease endemic countries. We have a global network established over the past 80 years. And of course, one of the key partners is indeed the Ifakara Health Institute, with whom we have a partnership in place since 70 years. I can easily say the Swiss Tropical and Public Health Institute would not be what we are without these long-term partnership built on mutual trust. So last year, as an example, we had more than 380 active projects in basically more than 100 countries, a lot of partner countries in the African continent, but also more broadly. And I'm just coming out of board meetings at the Ifakara Health Institute with the Director General of the Ifakara Health Institute. And I also salute him now from Bagamoyo. He remained in Dar es Salaam, but I want to really express my gratitude for the many, many years of very productive partnership. A few words about Swiss TPH, our strategy. We have established now our new 4-year strategy. It is all about improving health and well-being, and we do it through 5 really large topical areas. One, of course, as it is probably one of the greatest societal challenges, climate change and its impact on health. But today, what I will focus, and then particularly my colleagues, is infectious diseases. The big killer diseases like TB, malaria, and HIV/AIDS, but then also a whole range of more neglected tropical diseases. We have many, many other areas that we address, but the focus today will be on neglected tropical diseases and then tuberculosis. My colleagues, 2 of them are from the Department of Medical Parasitology and Infection Biology. And just to give you a flavor, that department works with many, many colleagues on malaria, but also tuberculosis. The focus later on will be on tuberculosis, but you see also a range of neglected tropical diseases, and more recently also viral infections. Of course, we have seen viral infections also during the COVID pandemic, but now also with outbreaks, re-emerging diseases, often based on viral pathogens. Now, let me focus on the neglected tropical diseases. This is what you had kindly asked me and invited to say a few words. The neglected tropical diseases, let's say this phrase or this construct came about approximately 20 years ago. And it was a very smart move by a couple of colleagues to then say there are a group of diseases, and if we put them together under that umbrella of neglected tropical diseases, that shows that they have a global burden that is probably comparable to tuberculosis or malaria. And that would then help also to raise advocacy, to raise a political movement to not neglect them any longer. And this was the initial list that was put forward in 2006. See, there were about 13 different diseases. And when you looked at them together, there was a global burden of approximately 50 million disability-adjusted life years, comparable to any of the so-called big 3 killer diseases, HIV, tuberculosis, and malaria. So again, this was a strong argument, political argument, to then not neglect them any longer, to move them forward. Today, the World Health Organization has an official list of 21 neglected tropical diseases, and it's quite a range of different diseases. They might be viral, such as rabies. You have bacterial diseases, such as trachoma. You have then also protozoan diseases like human African trypanosomiasis, and the largest bulk are parasitic worm infections, helminthic diseases, and later on I will briefly focus on 2 of them, schistosomiasis and soil-transmitted helminthiasis, both diseases, for instance, endemic in Tanzania over large parts. There are a couple of other diseases, and the latest one that came on the list is Mboma, really neglected, some people say the most neglected of the neglected. And that helps, again, with this list to then move research and development, control efforts forward. Latest figures in terms of the global burden of the neglected tropical diseases, maybe in the order of 16 to 17 million, considerably lower now than when we compare with malaria, tuberculosis. There was also major progress made over the past 10, 20 years, often with large-scale control program. I will give you one example later on. Now, in the remainder, let me give you 2 examples. One is on human schistosomiasis. Here I depict the life cycle of the disease. It's basically humans who are infected, they transmit and they excrete the parasite when they are infected, reaches open freshwater bodies, and it is the human behavior getting in touch with the water, that then they can become infected. So we already see by this life cycle that it's a disease that is closely linked to poverty. People who do not have access to clean water, improved sanitation, who do their laundry in open freshwater bodies, they would then be at risk of infection of the disease. So the social ecological context is important to understand also if you want to control, eliminate the disease. The most important strategy, the global strategy over the past 20 years has been the so-called preventive chemotherapy. What it means is that you treat people, often school-aged children, on a blanket approach, yearly, for instance, with safe, highly efficacious drugs, and often these drugs are provided for free by pharmaceutical companies. But of course, this is also then a strategy that is maybe not sustainable, because you need these control programs on a year-to-year basis. So if you really want to get to elimination, you need to improve social economic settings, but also improve water, sanitation, educate the people that they would not get in touch with the infected water. One example of how we can identify risk areas, and this is work that is done at our institute, in this case now specifically Konatsu and her group, is to utilize large-scale global databases to then identify what might be demographic predictors, environmental predictors, socioeconomic predictors, so to come up with risk maps and identify the high-risk areas. That's where we then can also intervene more precisely, not having a blanket approach year after year, but a more targeted, a more tailored approach. And what is quite interesting, if we look now at the global distribution of schistosomiasis, and sub-Saharan Africa is actually the geographical area where most of the cases occur, over the past 15 years, we have seen a reduction in the prevalence of infection by more than 60%. I would say this is one of the most effective public health programs ever. Within 15 years, a reduction in infection by more than 60%. This has now been established, the evidence established through Bayesian-based geostatistical approaches. I give you a second example to round up. Here I focus now on the so-called soil-transmitted helminths, parasitic worms that are transmitted either by walking barefoot or through the fecal-oral route. There are three main species, Ascaris lumbricoides, Trichoris trichuria, and the two hookworm species. It's a very simple life cycle. People are infected, and it's again their behavior, open defecation, and then through the oral route that people would become infected. Multiple areas how you can intervene, how you can break the life cycle, but the dominant strategy is again preventive chemotherapy, yearly mass drug administration to treat people and make sure that they would not develop morbidity. There are currently 4 drugs on the World Health Organization model list of essential drugs, and the 2 most important ones are albendazole, mebendazole, often given free of charge by the pharmaceutical company. Problem is that against hookworm and trichoris, the current arsenal of drugs do not show extreme good efficacy. And that's why we need to develop also novel drugs that have a broad spectrum of activity and a high efficacy in terms of cure rate and egg reduction rate. And this is where the Institute, this is now work mainly by Jennifer Kaiser and her group, have now established an evidence base that emodepside, a drug that comes from the veterinary sector, developed by Bayer, a pharmaceutical company in Germany, and we have partnered with them. And the essential work was now done in the island of Pemba to demonstrate that a single dose of emodepside already at the low dosage gives very high cure rate. This was published 2 years ago in the New England Journal of Medicine, and actually a Tanzanian PhD student was the first author, somebody from you at the Ifakara Health Institute. We are, of course, very, very proud and congratulate for this important piece of work. As a next step, then, phase 2b trial was done, again on Pemba, larger scale, and it could be, again, confirmed that emodepside at a single dose shows very good efficacy against both hookworm and trichoris. So it's a very promising compound, and the Institute, in partnership, is now developing this drug further. Now, to wrap up, I hope I could show you why we should really look at the neglected tropical diseases, because they cause a considerable global burden. And there are also tools available to control them, but the tools, the strategies need further refinement, need further improvement. For instance, novel drugs that show high efficacy against some of the soil-transmitted helminths. What we would hope is that in the longer term, a more targeted, more tailored approach comes along. As we move towards control and elimination of the diseases, we then need to identify where are the high-risk areas, which drug to use in which place, at what dosage, so that we have the best outcome with limited resources available.
All right, thank you so much. I think that was an incredible talk with lots of new knowledge. As we wait for more questions from the audience, please send your comments in the chat box and we will be reading those, a few of those in the limited time. But to pose the first question, given the current fluctuating rate of funding available across the globe, where does that put already neglected diseases? Who are the funders? Where do we start? Because the current toolbox for the diseases is already limited. So how do we continue to make strides as the funding continues to diminish?
It's a very important issue. We have already seen it during the COVID pandemic, when then some of the large-scale control programs could not reach the same coverage as in the pre-pandemic situation. People were able to catch up again, and now with all the turmoil in global health, there is a considerable concern that then the resources available might not be sufficient to really then target all those in need. That's where, again, I think an integrated approach and an approach where we can then tailor the limited resources to those people, to those communities at highest risk, makes a lot of sense, so that with the limited resources, we make most cost-effective use of it. But there is indeed a concern with now limited or funding in the global health scale, space, that then some of these neglected tropical disease might rebound.
And to really focus on the aspect of integration, at the intersection of One Health and soil-transmitted Helminthiasis, what is the risk and how can we leverage these two diseases that are really more or less based on our social and personal responsibilities? What is the risk and how can groups who are involved in this two not really distinct works join forces to be able to make some progress?
So I think the integrated approach comes there where we see with only a drug-only approach, mass drug administration year after year, it lacks sustainability. It is an important strategy to get morbidity under control, but in the longer term, we then need to supplement it with other tools, for instance, improving access to clean water, sanitation, raising awareness. It's then also the health education, communication, and educating people so that they change their risk behavior, and that is really important. You mentioned also the One Health aspect. There are some concerns that maybe there are also species that are in the veterinary sector that might also transmit diseases to humans. So we would also need to look at that issue. But in general, schistosomiasis or soil-transmitted helminths are specific to humans. So a One Health approach would not be the most logical one to consider compared to other diseases.
And lastly, from myself, what do you have to say about the role that neglected tropical diseases play in the not attaining of SDG goals in the Global South?
So this is, of course, all within the Sustainable Development Goal number 3, good health and well-being. And I would just in general say, if we are able to improve living standards by people, reduce poverty, so it's also strongly linked to Sustainable Development Goal number 1, and also education, Sustainable Development Goal number 4, we will have a major impact on the neglected tropical diseases in general. Whenever we see a country moving up in the social and economic development, we also see a decline of the neglected tropical diseases. So we cannot just look at the, let's say, at the neglected tropical diseases in isolation, but if it goes hand in hand with reducing poverty, improving education, and water and sanitation, this will have a major impact in reducing the neglected tropical diseases.
And lastly, I would ask you to speak to a young scientist who's interested in, or who's really been doing a lot of work in neglected tropical diseases. If you were to speak to yourself 20 years ago, what would you tell yourself?
I will tell myself, remain curious. There are some neglected tropical diseases where we still do not know how exactly the diseases are transmitted. And I would say that is unacceptable in the year 2024, with so much progress in cutting-edge research, that there are some diseases we still don't know exactly the route of transmission. So I would encourage young people, put your energy in neglected tropical diseases. That's where you can make a difference.
Awesome. Thank you very much, Professor Jürg. We will now take a 5-minute break while we switch the speakers for our next talk on tuberculosis. So please don't leave. We'll be right back.
Welcome back to the second part of today's Ifakara talk, where we'll be looking into current state of epidemiology, diagnostics, and research on tuberculosis. With me in the studio in TB research, Dr. Damien, Portevin, the group lead for TB Immunology at Swiss TPH, Dr. Klaus Reither, head of clinical and leader of clinical TB research group at Swiss TPH. And in front of me is Professor Sébastien Gagneux, the head of the Department for Medical Parasitology and Infection Biology from Swiss TPH, world renowned for his work on evolution and resistance for TB. Welcome, Professor.
Very much.
We will be having a series of 3 talks from Professor Sébastien, one from Professor Sébastien, and from Dr. Damien and Klaus later on. So please put your questions into the chat box, and Professor, please take us through the session.
Thank you very much. Also, from my view, my point, thank you very much for this opportunity to talk about tuberculosis here at Kingani. And we are extremely excited to be here and also share some results that were actually generated in close collaboration with IHI. And together with my colleagues, we agreed to coordinate our talks in a way that we're going to start with a brief introduction on what tuberculosis is and what some of the challenges are. Then I will go dig into some perhaps more basic aspects which are dear to my heart and the work that we do, more on the evolution of this important pathogen. And then Dr. Klaus is going to talk more about epidemiological and clinical aspects. And then we're going to end with Dr. Damien on the immunology, on some of the diagnostic challenges and some new way forward to diagnose tuberculosis.
Wonderful.
So that's a bit the menu for this afternoon. And so without further ado, let me just start with just some very brief introductions of what tuberculosis is and why we think, it is important. It's not just we think it's important. So you see on this slide, I just picked a few pictures which kind of illustrate some of the key characteristics of this, yes, we can say dreadful disease. It's one of the reasons I'm putting one of the figures here with the patient is that when some people don't really get to see tuberculosis patients that often, but it's also a way to illustrate some of the old names. Right? Tuberculosis is a very ancient disease, and history books are quite full of different names. And so you think of consumption, for instance, or the white plague. So these are some of these alternative names of tuberculosis. And in particular, consumption quite dramatically reflects what actually happens to patients when they are, they have tuberculosis and particularly don't get proper treatment. You mentioned antibiotic resistance, which is a huge issue in tuberculosis this evening. But again, the patients you're seeing here is actually someone who has TB, which is highly drug resistant. So almost, we cannot treat these patients anymore. So I mentioned antibiotics. You see that it's a bacteria, so that's different. It's not a malaria parasite. I always provocatively say we heard about the nice Schistosomiasis life cycles, very complex. You all know about malaria life cycles. TB is kind of boring from that point of view. It goes from human to human through the air, right? It's aerosol. You see that here also illustrated. It's an aerosol transmitted infection. So that makes it extremely successful and efficacious in terms of getting from one person to the next. So there's no environmental reservoir. It's a direct transmission. And what makes working with these bacteria complicated, it's because it's a so-called biosafety level 3 pathogen. So we have to take extreme high precautions when we actually work with these bacteria. And that makes the work quite complicated. So it's a deadly disease. One way to illustrate this is if you just count or estimate the number of people that actually have died of tuberculosis. So it's not just how many people had tuberculosis, but how many people actually died of it in the last 200 years. And see, it's an astronomical number, talking about a billion people. Huge number. So it's an extremely deadly disease, both from, let's say, more population point of view, but also from an individual patient point of view. And it's a bit illustrated here. So before the era of antibiotics, or maybe 60, 70 years ago, you cannot really properly treat this disease. And therefore, essentially, people would die in about 50% of cases. Fortunately, now we do have antibiotics, so these work. It's not quite straightforward. It's a complex cocktail that you have to swallow essentially every day for many, many months on end in order to get cured. But the good news is that generally it works. The problem comes with resistance. So you see multi-drug resistance, and then we also refer to extensive drug resistance. That's when these bacteria become more and more resistant to all these different kinds of drugs. And then you see case fertility goes back up to almost 50%. So some people refer to this situation as the post-antibiotic era, right? We're running out of treatment options, and that's of course true for many other bacterial infections as well, not just for tuberculosis. And this is the very latest numbers from the TB report from last year. So it's 10.8 million new cases every year. There's 1.25 million people who die of the disease, despite of being essentially treatable, which is actually very dramatic. It's actually more than HIV and malaria combined, and that's every year. I also looked quickly for the numbers in Tanzania. It turns out Tanzania is one of the 30 high-burden countries for tuberculosis. So it's a very dramatic reality also for this country here. 122,000 cases every year and about 30,000 deaths in Tanzania per year. You might wonder, what about Switzerland? So I also added the number of Switzerland. I often do that also to make the point that there's no single country in the world where TB does not exist, because that's often a misunderstanding, misconception, particularly people in Switzerland. We ask them about tuberculosis. What are you talking about? Tuberculosis has been eliminated like 50 years ago. I say, no, even in Switzerland, we still have TB cases. Yes, it's true. Many of them actually, migrants that come from places where there's still a lot of TB today. But just to make the point, TB is everywhere. There's a saying in tuberculosis epidemiology when you say TB anywhere, TB everywhere. Right? And it's maybe not surprising because it's airborne, right? So it travels with planes, it travels with people. Right? And that, of course, is an additional challenge. We heard about disease of poverty, or let's say, the poverty that leads to disease. Again, tuberculosis is a very good example. I often use this slide here to make an additional point. So if you focus on the left, depending on how it's projected, the green line, right? What you see is the TB incidence, so how much TB there is in a given country. So every black dot is a country. And then you have the GDP, right? So you see there's a direct negative correlation between, let's say, how rich or a country is and how much TB there is. So in other words, the poorer the country, take GDP as a measure, the more TB there is. You could also argue the other way around, right? It's a correlation. The more TB there is, the poorer the country. And that's actually an important point, right? We say, okay, yes, it goes both ways. Right? So TB causes poverty, and as a condition, you have poor people who are more likely to get tuberculosis. So I think it's quite a good example to actually illustrate this two-way interaction of how poverty can lead to health problems. And on the other side, you have essentially equivalent when you look at malnutrition. And I'm focusing on this. Of course, this is also a proxy of poverty, maybe not surprising, right? Turns out, I'm sure many of you have heard of the very tight interaction between TB and HIV. You know, HIV-infected individuals are extremely susceptible to tuberculosis. Turns out, if you look at the global scale, the most important risk factor for tuberculosis is actually undernutrition. So again, so malnutrition is globally the most important risk factor for tuberculosis. There are others, such as alcohol is used, smoking. Smoking, not a good idea, bad for you. It's bad for tuberculosis, bad for many other things. But in fact, it's actually a very important risk factor for tuberculosis. And then we have HIV. And another interesting feature here is diabetes. So type 2 diabetes, which is a classical non-communicable disease, has very close interaction with tuberculosis. This is something which is known as the double burden of disease, right? So you have in other words, people with diabetes, they're on average about 3 times more likely to get tuberculosis than people without diabetes. And there are places in the world, for instance, in India, where you have a huge epidemic of diabetes as we speak, which actually drives tuberculosis at the same time. So in India, for instance, diabetes is much more important as a risk factor for TB than, for instance, HIV is. Just to make the point, right, that it's a highly complex disease, and that's also illustrated here. I just showed you 5 examples of so-called risk factors, but it's very much a societal disease. Right? It's so many features and aspects of tuberculosis that we need to understand and, of course, act upon to hopefully make a difference in the future. And of course, we cannot do everything. An individual research group cannot do everything, and that's the reason we, in the past and still today, we're actually focusing on one particular aspect of this triangle, right? Because on the hand, we have the environment, we have the patient, the host, and of course, we have the pathogen. obviously you need a pathogen to cause a disease, but there's many other things, of course, also. And so why we became interested in studying this interesting pathogen is because, there's many reasons, but one of which is it comes in different flavors. So I'm showing here a family tree, if you will, of TB bacteria. If you go around the world and then you collect TB bacteria from patients and then you analyze them, you can genome sequence them, you look at the sequence of these strains, and you can build these so-called phylogenies. And then you see they fall in these different groups. And so I call it some. Currently, we know about 10 of these groups, and which cause disease essentially in humans. We earlier discussed about One Health in Schistosomiasis or Helminthiasis. In TB, there is also a component of One Health, because there are subgroups of TB strains, the ones in black here, which mainly affect animals, like cattle, for instance. There's the phenomenon, of bovine tuberculosis. So you can get TB also from cattle, but it's a different pathogen. So that's not the one we're talking about. Now we're going to talk about the purely human variant. And why is that interesting? Because when you look then around the world, you will see that these different human variants are kind of non-randomly distributed around the world. So you have places in the world where you have a blue TB, you have red TB, green TB, etc. So I mentioned this family tree of TB strains in different colors. So we talk about lineages, we can call them clades. And some of these are more frequent in particular areas of the world, and others are a bit everywhere. If you look at Africa, it's actually very interesting, because Africa is the only place where you see all of these different variants. And this per se actually indicates that most likely tuberculosis originated in Africa, because that's where you have most of that diversity. I'm going to talk about humans a bit later on. And we know that, and some of you might know that, the same is true for human beings, right? There's much more diversity in humans across Africa than in the rest of the world combined. And we also know that humans originated in Africa. And so humans, Homo sapiens, and then, left Africa to essentially populate the rest of the world. And we think that perhaps something similar is happening with tuberculosis, but we're going to get back to that in a minute. So this is essentially a hypothesis, which currently is supported by the observation that Africa also harbors the largest diversity when you look at these different TB strains. There's 10 groups I mentioned earlier on. Out of those, 6 exclusively occur in Africa, and 4 are a bit everywhere. Why this is perhaps relevant, we're going to discuss in a minute. When I mentioned about some of the specificities, right, that we see, there's a particular variant which only occurs. Well, a good example, for instance, is what you can see here is brown and green. You see, we only see it in West Africa. We don't know why. But we have one hypothesis, and one hypothesis is this idea of generalists versus specialists, right? That you have some TB strains might equally good at infecting anyone in the world, whereas others might be really kind of specially adapted to a particular group of people, right? So you can illustrate this like this. So you have a blue TB strain, right, which we call a generalist, is equally good at infecting and causing disease in blue people as well as in pink people. Whereas perhaps the pink bacterium, might be good at infecting pink people because it evolved with pink people for a very long time. But for some reason, it's not very good at infecting blue people. You might say, okay, why is this relevant? This we can discuss afterwards, assuming this is true, right? Again, we're talking about a hypothesis. Originally, we came up with this idea that this might actually happen. That's quite some time ago when I was a postdoc in San Francisco. San Francisco is interesting because at the time there was still quite a lot of tuberculosis, mainly immigrants bringing TB into the city, but then also TB transmitted. Remember, it's an airborne disease. So it started transmitting in the city of San Francisco. And San Francisco is extremely cosmopolitan, right? So you have people from all around the world. So somehow we could test this hypothesis a bit to see whether blue TB essentially infects mainly blue people. I'm going to tell you what blue people are in this example. Pink people, more pink people, etc., etc. And essentially, what we saw. So we divided the TB patients based on where they were born, which is about the proxy of what kind of humans they are, right? Let's call it in that way, mimic. And then we looked at what kind of TB they would get, right, and over a 10 years period. And that's what we refer to as sympatric. So TB strains were much more likely to spread successfully in patients that were somehow associated, geographically, where these TB strains dominate usually. And so kind of supporting this idea that perhaps this idea of generalist, specialist adaptation between humans, between these bacteria and the humans might be actually very true. So that's kind of a bit of, in terms of interaction, because then we say, okay, so can we look at this a bit more specifically, and why don't we do this in Tanzania? And that's where IHI comes in, the whole TB team in IHI, under the lead of Jerry Heller, Dr. Jerry Heller. So he was really instrumental in helping to set up what is known as the TB DAR cohort, right? So it's essentially a district hospital in Temeke where we have been recruiting, and essentially Jerry's team has been recruiting TB patients since 2013. So it's a long period. And there are many studies have come out of this. And I'm only going to pick 2 examples of some of the work that we did relatively recently in a joint project, which was funded by the Swiss National Science Foundation. Just finished a year and a half ago, and now there's other activities we're going to hear about afterwards. So this is past work, but I also would like to highlight that given, we're talking 12 years, right, that we have been collaborating on this work, there have been many opportunities also for training, and this is essentially what I'm showing here. So I'm sure you recognize many names here, be it, master students or PhD students or both, right? So some actually have worked on these different projects and did both of their degrees. And of course, we also had some Swiss and other nationalities benefiting from this long-term collaboration. That's actually very nice to see. The science is being done, but at the same time, people are being trained, and that's actually the people who do the work at the end of the day. So extremely proud of this as well. And on the Swiss TPH side, there were 2 people I really would like also to acknowledge. One is Dr. Daniela Brites, who is a senior scientist in the group, and Dr. Michaela Zwier, she just recently completed her PhD. And what I'm going to talk about now really focusing specifically of what we found in Temeke, when we focus now first on these TB bacteria. You might be curious if you remember what I showed you about these different colors, strains, right? So what kind of TB strains do we have in Tanzania? That would be the immediate question. And the answer is essentially we have 4 of these 10 colors. We have 4. Turns out it's the 4 variants which are the most common around the world. So unfortunately, perhaps we didn't detect anything specific, which we only see in Tanzania, only see in East Africa. Was perhaps a little bit disappointing, you might say. But nevertheless, there were some interesting features. So if you see what is labeled there as no, we then thought, okay, is there something we could look at this? When I saw origin of tuberculosis, TB comes out of Africa. Is this also true for what we see in Tanzania? And the short answer, essentially no. Because what Michaela did, she actually reconstructed the most likely origin of what we see today in Tanzania. And there we found something different, interesting, is that essentially all these TB variants that we see in Tanzania were actually introduced from outside, relatively recently. I mean, between 300 years and 20 years ago. So some very recent, some a few hundred years ago. And we did not find anything which perhaps has been here for thousands of years. So that was an interesting finding. Again, you might say, you know, is this relevant or not? I mean, this we can discuss afterwards. This I'm gonna skip because we don't see it properly. Where you might say, is that potentially interesting? Why is this particular variant I was referring to, the number 10, right, in purple, so frequent? Is it just because it was the one which was introduced the earliest, and they had just more opportunities to spread? And that's what's essentially shown here. So the hypothesis, the earlier you introduce, the more successful you become. That's the hypothesis. And essentially, that's not what we see. I mean, yes, in terms of 10, we see, but that's not the only explanation. Because if you are number 9, for instance, it's also introduced relatively early, but it's not as common. So it's clearly something else is going on. And that's when the humans come in. Right? Because remember this hypothesis I talked to you about from the San Francisco example? There we just took place of birth as a kind of a proxy. Right? But we can do better if you actually analyze not just the bacteria, but you actually analyze the humans, the patients. And that's where this human ancestry aspect comes in. And I would like to just briefly mention this because it's I would say very high-tech types of analyses. Now, we didn't find hugely striking results. I can already kind of spoil that one for you, but never, just to highlight that we did, you know, fancy human genomic analyses together with IHI, which was actually very interesting from that point of view. And that's why I wanted to briefly mention it. So it was really this idea of how these different variants, right? I mean, now we know we have 4 variants essentially in Tanzania interact with different human beings. And we cannot assume all humans in Tanzania are the same. No, right? We know they're not the same. But we also don't know exactly how different they are. Right? So that was a bit the question. Can we see perhaps there's some, now I'm talking genetics, right? Genetically different. How are people from Temeke, TB patients from Temeke, how genetically different are they? And could this perhaps influence the type of TB they would get? That's why I'm coloring the humans on the other side also in all kinds of different colors. And this, of course, we did not do alone. I mentioned that, again, the IHI was actually a 3-way collaboration. IHI, Swiss TPH, and EPFL in Lausanne also joined forces because they are the ones where the experts on genetics work. And we then also continued that collaboration. I just need to before I get to this human ancestry, it's a very short attempt to explain how human genetics works and how I'm going to refer to this as genetic segments. So you know, we will say human exchange genes, right? Everybody's familiar with this. Right? You know what I mean with this. You also know that you get some genetic information from your father and you get some genetic information from your mother. This is kind of put together, right? And that essentially makes you unique, right? And if you go further back in generations, you see, I mean, you got something from your grandmother, and presumably, and you got something from your grandfather and your great-grandfather and your great-great-grandfather. You can go back in time in all these generations. And you will have inherited some genetic information from your ancestors. But it's important to realize that maybe also not, because things can be lost on the way. Right? You don't inherit everything. And the further back you go, there's a chance that some of you say, my great-great-great-great-great-grandfather was the king of I don't know where. Doesn't mean that you necessarily genetically related, because some of the pieces of this genetic segment might have been lost on the way because you don't inherit everything. It's also the reason that child 1 here is not identical to child 2, even if they have the same mother and father, except for if they're identical twins. the reason I'm mentioning is this. You see, you also have colors here. So humans are colorful in the inside in terms of when we say genetic diversity. And so the point of these human ancestors is try to interrogate this inside, these colors inside, if you will. When I say inside, is the genetic composition of these different people. And that's what we did. So these TB patients in Temeke, we did not only look at the bacteria, but we also look at the humans. Now, this is also a bit difficult to explain, but I make it very easy. So we have 1,444 TB patients with the genomic information. It's raw, so every row, it's tiny columns, tiny columns, one after the other, which makes the 1,444 patients. And what you're seeing, these different colors are essentially representations of these genetic segments I was talking about in a very rough way explained. So you see there's a lot of black and a lot of green. But there's many other things also, but tiny proportions, right? And essentially, and that was a bit surprising to us in the sense that essentially the black and the green proportion of the genome of these people correspond to what some of the genetic characteristics that we know from people who speak Bantu languages. I say, okay, it's not surprising, right? Most of the languages spoken in Tanzania are Bantu languages, but it's not only Tanzania, as you know, with neighbor countries. So we were a bit disappointed, I said, because we were hoping to see more diversity, right? More colors, like bigger differences. Turns out that's not what we see. So in other words, the TB patients from a genetic point of view were actually quite homogeneous, more so than what we had expected. And perhaps not surprisingly, when we added the bacterial information, we didn't see any differences in terms of association. So that was all negative results, right? In science, sometimes you have negative results, and you also need to report negative results. So this is an example of this. No associations between the bacterial variants and the humans, no association. We also look at TB disease. Nevertheless, the work was done, and we learned something here. And this other part and goes straight to the conclusions. So I can leave the floor for my colleagues. So I just to finalize, current evidence strongly suggests that TB bacteria somehow originated in Africa, moved around the world, but what we see in Tanzania today most likely was reintroduced at a later stage. And that's what's dominating the TB epidemic in Tanzania and maybe also surrounding areas currently. Unfortunately, we did not find any specific interactions between the human diversity that we saw in Temeke and the TB strains. And the last part I could not talk about is that we think that these different TB variants actually differ in way they might be transmitting between people. But that's going to be perhaps for the next discussion. And with this, I would like to thank you very much. Funding agencies, thank you very much, of course, to the patients that participated in this study, the TB group at IHI, the TB group at Swiss TPH, our collaborators at UPFL, and you all for interest. Thank you very much.
Thank you so much, Professor. To the audience online, we ask you to be patient with us. We're going to take a 2-minute break so that then we can set up our next presenter. And then we will take questions at the end of all 3 presentations. Please do not leave. There's more interesting on TB. And at the end, we will have a wonderful time to just discuss more about this. Thank you so much, Professor. Again, welcome back. With me now is Dr. Klaus Reither,
Mhm.
Head of Clinical Research Units and leader of the clinical. Just briefly share with us about your career trajectory and experiences in TB research, highlighting one of the things that you think were most interesting that you actually worked on.
Okay. Yes, I would like to. I must say I started on a different end. I started as a radiologist and also specialized in radiology, but then I changed my way and went into tuberculosis research. And I think the most interesting part was really the time here in Tanzania. So Tanzania is my second home. I lived here for 7 years, 3 years in Mbeya and 4 years in Bagamoyo and Dar es Salaam working for IHI. And so these were the time where I really saw firsthand the problem of TB, and we could really establish clinical research in both sites in Mbeya and here to tackle the certain problems with regard to TB, diagnostics, treatment, and vaccine.
Thank you.
Yeah. And I must say it was the best time. Of course, now I enjoy being at Swiss TPH. My director is sitting here, but it was the most adventurous time. So.
Welcome, the floor is yours.
Okay, so I would like to talk about TB and especially about asymptomatic or subclinical TB. And I would like to do this to also convince you that this is a very important topic, a topic which was, we heard the word neglected by the TB community for a long time because it was not known, and its significance is quite substantial, and it will make a big impact to really address this problem properly. So it's more an overview on a certain aspect, but you will see it's a big, large proportion of TB in the world. This is just the agenda. I don't have to go through this. So here you see the global TB incidence in the past 13 years. And what is really striking is that the progress in terms of numbers of incident cases is really not substantial. It's even going up in the last years. You can see that the confidence interval is getting a bit smaller. So we are more confident that these numbers are correct, but this is not a real progress. And I would say there are many factors which play a role here, but subclinical TB or the ignorance about subclinical TB plays a big role why we are in where we are at the moment. I first would like to go a bit back to how we have learned about tuberculosis. Most of you might have learned that there is active TB, which is now the tip of the iceberg in this picture here, and latent TB, which is the body below. And there was, for those who have latent TB, a lifetime risk of about approximately 10%, and the TB infection is actually lifelong. That's what I learned, at least in medical school. Meanwhile, we know that the reality is quite different. So TB is actually a disease which has a spectrum of different stages. And these stages, the patient can go through in both directions. So it's bidirectional. And you see one of the most important parts here is the so-called subclinical TB. I will come to the definition later. But this is a stage where the patient doesn't have symptoms, but has already microbiologically confirmed tuberculosis. There are many other models, meanwhile, but they're all, in essence, as it is shown here now, show the same thing, that this is a dynamic disease spectrum. So it's not only latent TB and active TB, there are different stages the patient can go through and also move between these stages. So what exactly is asymptomatic or subclinical TB? I use interchangeable because in the past it was always subclinical TB, and last year the WHO has called it asymptomatic TB. So there's literature on subclinical and on asymptomatic, but it's actually the same. So here the most common definition is that it's a disease due to viable mycobacteria, and it doesn't cause any clinical TB-related symptoms, but you have abnormalities which are detected by microbiological assays, and in this definition, even by radiological signs. That's a bit doubtful, but this was the most common definition. Then there was, people came together, a so-called Delphi consensus, and they looked at this a bit more closely, and they made a very nice definition, which is shown here, just to highlight the different categories. It's either non-infectious versus infectious, and then they look at macroscopic pathology, and then symptoms if present and not recognized by the patient. This led in the end, as I said, to the WHO definition, which is shown here. And, you know, the reason why I'm explaining this is not only because it's such an important part of the TB, but it's also the topic of our next larger project, which will start very soon. TB, and then this study here, which was presented in 2021, which was really an eye-opener. They have looked at prevalence surveys from Africa and Asia and found out that in the median, roughly half of the participants have subclinical TB, meaning they have TB, have microbiologically confirmed TB, but they have no symptoms. In other diseases like malaria and so on, this is commonly known, and it is, but for the TB community, this was really new. And I will later explain a bit why this is such an issue. And now I looked at the definitions, and really definitions matter here, because when you define subclinical TB as no persistent cough, then it's even 82% of TB cases. If it's no cough, it's still above 60%, so 62.5%, meaning the participants have no cough, but TB. I mean, this is really contradicting what we have learned so far. And if there's no symptoms at all, you still have around 1/3 of the participant or of such prevalence surveys. So this is, of course, for the patient very problematic, because the patient has a disease but might not experience now any symptoms, and the disease might progress. But on the other hand, there's also the question, is it a problem for the community? Do they transmit? There are not so many really data, prospective or retrospective data, but there are modeling data which are quite convincing. For instance, this one here from 5 cohorts and prevalence surveys they were looked at, and it says that approximately 35 to 51% of future transmission, so transmission in the next 5 years, comes from smear-positive subclinical TB. That means there is indication that this disease stage contributes massively to transmission. This is also supported by this study. It's again a modeling study, and they came even to the conclusion that 68% of global transmission is due to subclinical TB. Of course, they have a very broad confidence interval here, but even if it's a bit lower, it's alarming numbers, because these participants have, or these patients have no symptoms, are not visible in the community, they don't go to health facilities, but can spread the disease. There is one study which also provides some empirical data. It's coming from Vietnam. This was a combined TB prevalence study and a tuberculin survey. So tuberculin skin test is an indication of infection. And they showed on one hand that children living with individuals with smear-positive TB have an increased TST positivity risk, which is expected. But in a certain age group, so 6 to 10 years, the TST positivity risk was the same for those living with clinical TB and subclinical TB. It's already now a real-world indication that transmission takes place from subclinical TB cases in the family. Now, how can we then identify these people? How can we find them? Our whole system is based on passive case finding in tuberculosis. We wait in a health facility that people come to us with certain symptoms, and we try to rule out or diagnose tuberculosis. But this will not work in subclinical TB. So we need other measures, other methods to identify these people. And it's now common understanding that the best way is to do symptom-independent screening campaigns, meaning screening certain population groups, for example, targeted screening in certain population groups, irrespective of symptoms, because symptoms doesn't help us to identify subclinical tuberculosis. The idea is to reduce the infectiousness, to reduce transmission, to come to earlier treatment, and this leads to less severe disease and less sequelae. And of course, the financial consequences for those involved, individual and community, are diminished. So next question is, so which methods can we use to diagnose asymptomatic tuberculosis? And one of the most commonly used now is really digital chest X-ray, and often in combination with computer-aided detection for tuberculosis, which is a software which reads the digital X-ray and gives a probability score if or if not there is tuberculosis, but it also requires a second step as a confirmatory test. You cannot build a diagnosis only on digital X-ray. So there was a lot of hope in aerosol detection through face masks. There was a group who had very promising results by using face masks they have produced themselves. But we and other groups used those, not those face masks, but industrially manufactured face masks, and we could not reproduce the results. So this, at the moment, is not an option to be used. And now what we also will employ in our next study is tongue swabs. So tongue swabs is a very accessible non-sputum sample. And there is indication that it might be not as accurate as sputum sample, but it's much easier to obtain. And this comes down to the principle of diagnostic yield. So that means the population you want to examine, and when you have an accessible non-sputum sample, you might reach more positive cases than with sputum sample, where you have a smaller group who at all can produce sputum. So that's at the moment where we are standing, and Damien will talk about tongue swab more in detail. Next question, which needs to be asked, is how should asymptomatic tuberculosis be treated? It's a bit like we are addressing this, all the things we have already guidelines for, to half of the patients now, which were not known so far. And in these guidelines we have at the moment, there is no direct mentioning of asymptomatic tuberculosis, but there's a clear guideline: when you have microbiological confirmed TB, they should be treated. The problem might be, and this has also been shown in a meta-analysis from prevalence surveys, that all these people got standard treatment and success was comparable to clinical TB. The problem is, however, that the patient doesn't have symptoms. TB treatment is long, it has adverse events, and it is burdensome for somebody who feels well. So to take drugs, in combination drugs, for 6 months is quite a burden. So the idea is now to find out where exactly should be the treatment of asymptomatic tuberculosis. You have on one hand, on the left-hand side, preventive treatment. These are, in essence, except for one very short treatments now, we have them. And on the other hand, you have standard treatment. We have two different ones, 6 months, and now the newer rifampicin-based 4 months treatment. And maybe the asymptomatic tuberculosis treatment is exactly in between somewhere. And this needs to be determined through clinical studies and clinical trials. One of these trials could be, and that's an idea we have, probably a potential pragmatic clinical trial to do a so-called stratified treatment. On one hand, you have asymptomatic and mild tuberculosis with a shorter 4 months treatment, and the more hard to treat active tuberculosis with 6 months treatment, which is supported by other studies which showed that this stratification is useful in tuberculosis. Now, would the patients accept such a treatment? I said already, the patient has no symptoms, and there are very few data only available. But there's one study, the so-called Vukusatsi study, which was a study with high rates of asymptomatic tuberculosis and showed that HIV status had no influence on linkage to care. And they could show that the attendance, or let's say the cascade of care, is quite similar to between asymptomatic and symptomatic, meaning that the patients complied with the treatment. Of course, it was in the frame of a study, not in a pragmatic setting, but I think this is already one indication that it could work, that people really complete the study, even if they don't have symptoms. And now the last topic I would like to touch is TB vaccine development. Unfortunately, TB, everything is a bit more complicated, I must say. And TB development is at the moment seen as the biggest game changer. When we have a good vaccine, then it should really change the prevalence worldwide. But also here, our main categories, prevention of infection and prevention of disease, have to be thought through again. But because when you look at this, like the cause of disease, infection, it's very variable, and the pathways through these different stages is very variable, as shown in this model. And that's why people now came together on TB vaccine development, and they now declared prevention of clinical and asymptomatic TB a priority. And they have developed different study designs, especially for prevention of disease studies, to address also asymptomatic or subclinical tuberculosis. So I would like to summarize what I hopefully could explain in these few minutes. So there is a huge epidemiological and clinical significance of asymptomatic tuberculosis, and it's really surprising that it only became apparent in the past few years. We need a standardized definition of asymptomatic tuberculosis for clinical control and research. That's essential. So we know that every second person with microbiologically proven tuberculosis has no symptoms. It's a totally new information. The mathematical model show that there is potential for transmission and that there is a heterogeneous cause of asymptomatic tuberculosis. And the detection strategy should focus on symptom-agnostic screening methods. These are expensive, these are laborsome, but there's no other way to identify people with subclinical TB in the community. And we cannot wait at the health facility that they come and present. The treatment of asymptomatic tuberculosis could possibly be improved by a stratified treatment approach, by a shortening of approach, but this needs proper clinical trials. And the vaccine trials should or will include asymptomatic tuberculosis as an endpoint. with this. Thank you.
Ooh. Thank you so much. Thank you so much. I hope with the audience online that they have seen some of the questions that were being displayed on the slides. I think that these are good opportunities for people to actually go and try and research into. So I think this is really, it's been a really, really informative talk, especially for me who's an TB person. Like, so there's so many questions that are out there that need answering just for asymptomatic TB. So thank you so much. We will take another short break as we set up slides for Dr. Damien. We hope that the online audience is still on, and if you have any questions, please do post them, and we will ask them at the end of Damien's presentation. Thank you. Ooh, welcome back to our last presentation from Dr. Damien. Welcome, Dr. Damien. I know you're the last to present, and I know your presentation would definitely be the most interesting, just solidifying what we have learned from Professor Sébastien and Dr. Klaus. So the floor is yours. Welcome.
Thank you, and asante sana. So I'm also very deeply attached to IHI because I think I've belonged as many years with the Swiss TPH than with IHI. I even started working at IHI before even stepping into the Swiss TPH. So as you will see, and I will acknowledge this very tight interaction between our two institutes and all the students who are also involved in this project. I have the difficult task to explain a complicated topic for a complicated disease. So I will try to do my best and explain you a bit what we have been doing and what we plan to do indeed in this subclinical TB study. So just as a little recap, this is the NTB partnership slide showing a bit how bad the situation is about tuberculosis. Sébastien has already very well explained 10 million cases every year, 1.25 million deaths. This is totally unacceptable. This is a curable disease. We have antibiotics, so this should not happen. And one of the reasons why it happened is because we don't have good diagnostics. And that's what brought me to collaborate initially with Dr. Reither to develop a non-sputum-based diagnostic, a blood-based assay together with IHI, and later on move on into other molecular-based assay. But from an immunological perspective, the best tool to control an infectious disease would be a vaccine, right? And as you may know, we don't have a very good vaccine against tuberculosis, so there's really a need for a better one. The problem is, if we want to build a good vaccine, we need to know what to develop in a human body to protect against this infection. And it's not very clear what takes a good immune response against Mycobacterium tuberculosis. And the reason is also overlapping what Dr. Reither has very well explained, is that if you see this green individual is an healthy individual, and if he's exposed to a bacilli through aerosol, there are multiple outcomes. We know that a large proportion of individuals will actually resist the infection. They will not develop any symptoms. They will clear the infection totally. We've heard that some will actually might remain infected but do not display symptoms. And at the end of the day, only a fraction, maximum 5% of people who are exposed to Mycobacterium tuberculosis will develop symptoms. So you see already our immune system is not equal, and there are risk factors associated with this immune suppression, HIV, of course, but undernutrition, etc. So to understand better protective immunity, we study in the lab macrophages, because macrophages are the main cellular host of Mycobacterium tuberculosis. Those phagocytes, they are here to clear pathogens, bacteria. Well, MTB benefits from these cells and replicate within these cells. So the very same cell which is supposed to clear the infection is actually becoming the house for MTB replication. So you can imagine the challenge from an immunological perspective. And then, so the human response says, okay, if I cannot handle myself, the macrophage will, this infection, the macrophage will seek help and lead to the formation of this so-called granuloma. So granuloma are just balls of immune cells surrounding the bacteria, trying to encapsulate, make it suffocate, and hopefully in a beneficial manner. But again, we don't really understand all of this, but we develop in the lab an in vitro granuloma tool, and I will get back to it. And we have developed together with it very advanced cutting-edge platforms such as time-lapse fluorescence microscopy, but also a very high-dimensional new cytometry approach, which is called mass cytometry, and very recently a single-cell transcriptomics approach, also generating very high-dimensional data. Now I want to just show you these very nice videos that we have recently acquired at the Institute that shows you in a dish, you have here macrophages plated sticking to the dish. And you see these red spots are Mycobacterium tuberculosis expressing a red fluorescent protein. And here you see on the right-hand side the bacteria being sniffed by the macrophage, and now the macrophage is attaching to the bacteria is engulfed, and we can basically follow in real time the interaction of the bacteria and the phagocytes. And this is really amazing to watch under your own eyes. This is, of course, within BSL-3 facility, so quite advanced technologies. And the cool thing is, this is one very specific foci that we can follow in time. This is the topic of a PhD, Yasmin Albi's in the lab, that we can quantify each individual bacteria within each individual cells at different time points. And what I want to show you here, you see each individual dot is the amount of fluorescence we detect in these macrophages across different donors, A, B, C, D. We can count the number of dots, and we can follow the proliferation of these bacteria. And what you can see is, depending on the donor you use, you see more or less replication. So there is inter-individuality differences in this MTB susceptibility at the macrophage level. But the most striking is that, first of all, this is a control experiment, because the standard approach when you quantify bacterial load in such assays by lysing the macrophage and plating and counting by hand. And we see that these fluorescence really nicely correlate with bacterial counts. But what is very interesting here, and I just depict 4 examples. So this is the follow-up experiment over time, the behavior of a bacteria within an individual macrophages. And you see 2 flat lines. So in these macrophages, the bacteria is not replicating. You see a bit of an intermediate line where the bacteria replicates, but in a slow manner. And you see this very dramatic growth. So not only individuals are different, but within the same individuals, cells also handle differently Mycobacterium tuberculosis. So this is a really brand new area of research. There's already some evidence in mouse, but basically we're doing this in human. And of course, we wonder if this implies or is the cause of this increased susceptibility of some individuals compared to others. As I said, we also build this 3D in vitro granuloma. So this is a bit more complex. So out of the blood of a given individual, you can isolate all the immune cells, the PBMCs. You infect them in vitro in the lab with Mycobacterium tuberculosis, and within a week, you see this granuloma forming by themselves. It's also nature, it's magic. You don't have to do much, just give them the right environment. It's in 3 dimensions, so we give them a matrix so they feel like as if they were in the lung. And what is interesting is we can study the bacteria. And what happened is these bacteria, initially, they're all green. You see in this fluorescent microscopic picture, but they can switch to a dormant phenotype. So they defend themselves by going into a sleeping mode, which we call dormancy. Of course, in this mode, they can wait to resuscitate many years later. We know latent TB can reactivate later in life, but it also renders the bacteria more tolerant or resistant to antibiotic treatment because they just sleep, so they are not susceptible to antibiotic treatment. What is very striking is that we performed this granuloma experiment from the blood of patients which were healthy. So this is benefiting from one of the very first study I did with Klaus Reither, a diagnostic study where we collected the PBMC from healthy controls here in Bagamoyo and TB patients. So we built in vitro the granuloma from these healthy controls, and we compared them to the granuloma of the TB patients. And we recovered the bacteria in these granuloma. So you see each individual dot here is the data from a given individual. And you see that the TB patients, for some reason, their cells tend to promote dormancy. So the bacteria enter dormancy in a more rapid way. So that was very striking. And we wondered, okay, what is special about these granulomas? And you don't know where to start from. PBMCs are quite complicated. There are many things to look at. So the beauty of the tools we use is called cytometry by time of flight. I mentioned it earlier. This is really something that allows you to look at many markers compared to a regular flow cytometry, 43 markers. as a combination, you can come up with thousands of different combinations for these markers, but we identified about 700 cellular phenotypes. And to make a long story short, we just looked at what is enriched in granuloma of these TB patients, and we depicted some cellular subsets which are all cytotoxic cells. And when we deplete these cytotoxic cells from the PBMCs prior performing the in vitro granuloma formations, we see that now dormancy is actually reduced. So really those cells are the one responsible for this dormancy phenotype. And we wondered, okay, what's going on? And it is quite pretty known that cytotoxic cells have in their cytoplasm granules, which are called cytotoxic granules, which are normally fusing with the membrane to be delivered to an infected cell. And we know that those granules have the capacity to kill the bacteria inside an infected macrophage. So you see the nature is really well done. But we knew that the granulizing this compound, which is present in the vesicles, is able to kill bacteria, but it was never envisaged that actually bacteria could resist this lethal activity and dormancy. So what we did is that we actually forced these cytotoxic lymphocytes to degranulate, and we recovered the supernatants of those cells, and we exposed the bacteria to these supernatants. And when you do this, you can actually see that you can induce artificially dormancy. So we could really demonstrate the role of this cytotoxic molecule. So you see that coming from really a clinical angle, a relevant specimen, we can really understand what what immunity needs to actually promote a given phenotype. Whether this is a good thing or a bad thing, we can discuss later, but this is really interesting. I'm coming back on these different flavors of Mycobacterium tuberculosis that Sébastien has talked about. These strains which are, for some reason, less virulent, it's hard to grow them. They seem to do not transmit so well, yet they persist, they are there, and it's difficult to understand how they are not outcompeted by modern lineages. So we decided to embark on trying to understand how the virulence of these different strains in these granuloma models. So we did the same again. We infected this time PBMC from healthy controls, and you see here these nice pictures showing the granuloma only in the case of infections. It's a tremendous amount of work. We have about 6 × 318 strains here across 4 different donors. This is the work of Ainoa in the lab. And you see here each individual dot is the outcome of 1 granuloma infections by 1 given strain. And we could reproduce this increased virulence if we can assume that replication rate is a virulent trait, but modern strain seems to replicate faster in the blood of the 1 given individual. So what I'm trying to say here is that I showed you already at the beginning that there is heterogeneity in the way macrophages respond to infection, and here I'm telling you that also the way Mycobacterium tuberculosis promote disease is very variable depending on the strains you're talking about. So when you want to design a vaccine, you need to take that into account probably. And going back to our dear interaction with Ifakara Health Institute, we've had this TB-DAR cohort that has allowed us to realize that this lineage 1 is highly prevalent in Dar es Salaam, and yet it is not outcompeted by these other modern lineages. So there's seemingly a strategy, a virulence strategy here specific for lineage 1 to compensate for this low virulence that we detect in vitro. Which brings me back to what Klaus has talked about, these subclinical TB forms. I don't have to explain this graph again, but technically there are as many people walking around without symptoms, harboring bacteria, than patients going to the clinic because of symptoms. So that's where we decided to maybe hypothesize that these different bacterial traits might be driving the subclinical TB, and we'll get back to it. And there's this difficult, let's say, a relationship between this reproductive number of a given strain. Reproductive number is the number of secondary cases that an index case may have when it exposes other individuals, and its virulence in terms of the number of deaths that these infections will lead to. So you can easily imagine that if you are highly virulent strains and you kill everybody very fast, it's the end from an evolutionary point of view. The pathogen has no interest to do that. So it's trying to balance this capacity to transmit, but not to kill its host too fast. And when you calculate this reproductive number, it's just a multiplication between 3 factors, which is the probability of infecting an individual, the average time these two individuals are in contact with each other, and the duration of infectiousness, if you're infectious for 2 days or for 6 months. So now you can easily see where we're going. The idea is that if you are capable of walking around for a long time while being asymptomatic, because you are not highly virulent, well, you increase your chance to induce secondary cases, so you compensate for this loss of virulence. And this is exactly what we want to demonstrate in this sub-TB study, that maybe there would be an increased incidence of lineage 1 infections over lineage 3 in subclinical TB patients. So how do we plan to do that? We thought the easiest way to find subclinical TB cases would be just to follow up index cases and their family, their relatives, the people that have been exposed. So we would like to recruit household contacts. And we came up with a quite ambitious number of 10,000 and try to screen them in a, let's say, cost-effective manner. And as Klaus has already explained, that this is tricky. But this is the work of Helminthiasis that I'm sure you know, where she really nicely developed a very efficient tool that can accurately diagnose TB within 24 hours. This is where the immunology comes into play. Unfortunately, even though this test is really fantastic and probably has a very good perspective, it's probably not suitable for a high throughput method. And so even though it was really delivering fantastic results, we thought maybe we should find an alternative. And that's when Mohammed and Yves have performed this study based on a urine screening. This urine is very easily accessible. We could even pool and screen a whole classroom. And the reason behind is that whenever you have an infection in your body, the lymph drains whatever is being killed, and that ends up in your bladder, and it's ultrafiltrated into your urine. And therefore, there are bits of DNA from any infectious agents, even your own DNA at the end of the day, that you can retrieve from a urine sample. One limitation is urine is highly diluted, it's full of water, so it's hard to take 5 microliters to perform a PCR. So what we did was a very clever approach developed by another lab, is to use magnetic beads which can trap selectively because they are bound to a probe which is complementary to the DNA sequence target of TB. And you can basically enrich for MTD DNA in such a capture methods. That was brilliant and actually delivered very good results as well. But on the way there, we realized it was also very technically challenging. It's not so cheap. So again, to screen 10,000 households, we thought maybe that's not the best idea. So then came the idea of using face masks, as Klaus also explained. And I'm making a very long story short here. Mohammed has tried, but with 25% sensitivity, we thought, okay, that's not the way forward. So now we're trying these oral swabs. It seems that this tongue is capture anything you breathe in, you breathe out, and the tongue is full of pathogens, including Mycobacterium tuberculosis. We have developed together with Mohammed and Josie a way to do a very quick and dirty processing of those tongue swabs, pure sonication, boiling of the samples and qPCR. And I should say we're quite happy. It's not perfect, but it really meets the criteria for our triage test, and I think we'll move forward as soon as the study protocol is approved, because we are still at the ethical stage right now. And let's get back to immunology. This is my last slide. So then what brings also the immunological component in that study is to maybe appreciate that not only lineage 1 might be driving the sub-TB epidemic, at least in Dar es Salaam, but potentially also the way individuals react to these infections may be also contributing to a better control and a lower appearance of symptoms. So in other words, and that's what I'm wording here, is can subclinical TB be driven by specific immune responses? This is the work of Lara, and also in close collaboration with EPFL. And we are here again aiming for a very ambitious approach. So we would like to compare these ex vivo granulomatous response from subclinical TB patients, regular TB patients with symptoms, and also healthy controls, and dissect them by single-cell RNA sequencing. This is really a beautiful technology, but it generates a lot of data. Long story short, you have such a slide, you have 120,000 wells, micro wells, in which the cells would fall one by one, together with a bead, which is able to capture the RNA of the same cell they are together. This is what you see in this picture. So once you add your lysis buffer, the RNA are immediately captured, and you can levitate the magnetic beads. Then, of course, they are it's tagged, right? There are some ways to, clever ways to assign the reads to these cells, to these beads. And then you sequence everything and you generate billions of reads. So we have done this already. We have established the methods now, sequence about 126,000 transcriptomes from about 12 TB patients and 12 control. 1.2 billion reads, it's tremendous amount of data, very much work in progress. I'm just showing you here a teaser on some areas of interest that we think are popping up, but I cannot tell you more today, and hopefully next time we are invited, I will reveal all about it. With this, I think I gave you a very broad overview of all our activities that we are developing together, and I would like to thank all the people involved, as well as the patients, of course, without whom we could not do any of this research. Our collaborator, of course, at IHI and also at Novartis, we give us in kind access to this site of platform, and the Fonds National Suisse for giving us all the money.
Thank you so much. Again, I think for those who work in TB, it's really, really interesting with the development of the diagnostic tool. I think that's one area across all research in diseases that we really need tools that are quick, fast, that you can really screen people and have high sensitivity, high specificity, so that then we can really be able to know the burden and how to really, put up ways to control this disease. Now, with that as the end of our final presentation, I would like to welcome back Professor Sébastien and Dr. Klaus to join us here. And then I will take some few questions from the online audience. And I would like to begin with this first question. Income countries. This is because we are fighting the TB war on multiple fronts, that is both from the pathogens and poverty. So what do you think our governments should focus on to have a big impact and not to have just a trickling effect?
Yes. But you have to remain optimistic, right? And I think it's a very good question in linking TB to poverty, but the same is true for malaria, for schistosomiasis, etc. I mean, we're all working on these diseases which are largely driven by poverty. So you might say, you know what? I'm not being provocative now. Just forget all this science. I mean, just focus on reducing poverty, and then you get rid of everything. Right? And there's some support for this. I mean, I didn't get the chance to show this, but there are some famous graphs. You can Google them. If you look at TB mortality in Europe, you would find all these dramatic graphs where you see how the TB was going down in the early 20th century, 19th century, let's say 20th century, long before any particular interventions. I mean, even before TB drugs were there, TB was already going down in Europe. And that's the reason, of course, if you ask people in Europe, oh, TB, what are they talking about? I thought it was they don't remember that. Maybe their grandfathers, they might remember, oh, yeah, we used to have TB in Switzerland, which is true. Big problems of tuberculosis. So that actually illustrates what you can achieve by reducing poverty. I mean, economic development per se is going to have a huge impact. And then the question is this going to happen during my or our lifetime? Then I can say just, yes, hopefully, but it's not sufficient. And so we need research, we need implementation already of existing tools, of course, right? I mean, there's things we can do with the tools that we have, but we heard about vaccines. I think that it's unlikely to happen, that would be my guess, if we don't get a better TB vaccine. I think the same might be true for malaria, right? And malaria is more advanced now. We have a very promising vaccine. It's not the superb final answer to malaria, right? But it's definitely going to contribute. And so we need something similar for TB.
Wonderful, wonderful. And a question for you, Dr. Klaus.
Yeah.
So this is from Michael Casimiri. He says, did the use of 3HR TB/3-HP give similar results to the standard TB treatment. This is for the, I think, for 6 months.
The 4 months.
Yeah, for the 4 months or 6 months.
No, we have two standard treatments. One is a 6 months treatment, and one is a 4 months treatment, which has rifampicin and moxifloxacin. And they have, this was the first non-inferiority proof of 30 years of clinical trials. That was the shortening of the drug regimens. So we have now two different ones. One is for 6 months and one for 4 months. And what I wanted to show is a bit that there is evidence that the longer ones is more effective or should be better used for hard to treat participants or patients, while the shorter ones can also probably better be used for those who have a low bacterial load or other disease, less disease severity. So these two we have at the moment as standard treatment.
And then there's a general question here to all of you three. What is happening in the frontier of TB drug research? Are there new technologies such as nanoparticles that can improve efficiency, reduce dose and side effects? And this is from our Director of Science, Dr. Aliolud.
Good question. Yeah, we can say that in the past now, quite a lot of advancements have been made in terms of new drugs. And I think the biggest advancement is actually the introduction of a new regimen. We had before XDR, pre-XDR and XDR treatment had a success rate of 80 to 90, a failure rate of 80 to 90%. More optimistically, it's really great that we have this. But on the other hand, we see already resistance occurring all over the globe. And we have a problem with bedaquiline resistance already now as we speak. And this is something where the diagnostic and the surveillance is lagging behind. I think there's one lesson learned is always how early you introduce a drug regimen, while the surveillance system is not in place always. But on the other hand, you also save a lot of lives by introducing it. And I think it's a very difficult question to answer how this should exactly be. And of course, as Sébastien said, if there were more financial means and there were more money involved, we could introduce both much easier.
Wonderful, wonderful. So,
can I follow up on this? I mean, I think it's important to realize also that these new drugs that so bedaquiline, Damien, that have been developed, it was the first new drugs in 40 years, right, to come along to treat these, particularly these highly drug-resistant forms of tuberculosis. And because now resistance is already emerging to new drugs, does that mean we're going to have to wait another 40 years for the next new drugs? Hopefully not, right? Because otherwise we're going to run exactly to the same problem again. So that's kind of the bad news. The good news is that there are actually even more compounds, you know, at lower, earlier stages of development. So there's hope that even if resistance to this new drug now is emerging, there will be other drugs coming along the pipeline. Keeping in mind that we need 4 drugs, right? It's not good enough to just come with one new drug. You need to, I needed 4 drugs to treat these patients. Otherwise, you're going to have resistance immediately. And that's actually, unfortunately, I mean, that's again the good news. The bad news, because we talk about vaccine, right? We didn't talk so much about vaccines, but we desperately need vaccines. And there it looks much bleaker, fortunately. So yes, there's one candidate which looks very promising. 50% efficacy in, you know, based on small number of data, which is now going into phase 3 trials, huge amounts of money being invested. There's not much else going on, unfortunately. In particular, up the pipeline, it's pretty empty, unfortunately. So if this one would fail, which there's certainly likelihood it might, then there's not much coming up the pipeline, unfortunately, which is another big problem.
I know we are pressed for time. Oh, please,
I have
please do.
something
Please
to
do.
add. I think another component which hopefully opens nice avenues, and Klaus, and you will probably complement, are the host-directed therapies, because tuberculosis, even if they are cured, post-TB life expectancy is reduced because lung function are often lowered. And nowadays, there's a lot of research aiming to improve the recovery of the lung while we get rid of the bacteria. It's not only getting rid of the bacteria, it's also about restoring proper lung function, which is often a very big problem for TB patients. And there are many trials ongoing in that direction.
Wonderful. I know we are pressed for time, but I have to give favor to Heleniza because she's asked a question directly to you, Damien. And so she is asking, what's the progress of HTT in TB? Is there an idea of more or less how the bacteria behaves in macrophages and granulomas? Can HTT be the magic bullet we need to attack MTB in the cells?
Well, I kind of just touched upon a bit this topic. I don't think there's a magic bullet as of today. There was some hope with vitamin D, for instance, supplementation, which seems to work, but only for people who have specific genotypes, so there might be benefits there. And vitamin D actually acts on macrophage functions to restore macrocytal functions. Metformin also, I think, was a promising target. I think the first data are not that great from what I've heard. I think it's still too preliminary to say. So I'm not aware of big progresses or yes.
Wonderful. That brings us to the end of our session. Please, for the audiences online, I know there are more questions. We will definitely send them to our guests and they will do written responses, and we will share this with all of you. And with that, we say thank you to Professor Jürg and Professor Sébastien, Dr. Damien and Dr. Klaus for your wonderful time here with us, for sharing your intellect and views on how the NTDs as well as TB and research is ongoing. We will be welcoming you again here for other sessions as well. And with that, we would like to say good evening, good afternoon, and good morning from wherever you're joining us.